Hello, very good afternoon to the student who have joined. Can you please let me know in the chat box if you can see the presentation? And also let me know if my voice is audible to you. Kindly confirm in the chat box if you can see the presentation. Okay, thank you so much. We will wait till it's 4 p.m. Then we will start today's session. Okay, so it's already 4 p.m. I think I will start with the introductions. Mm, okay. Okay, so very good afternoon. My name is Mousumi Deb. I am a PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I am your TA for this NPTEL course of Medicinal Chemistry, and today it is our week 6 live session for this course. So every week we discuss that week's course content via MCQ questions. So today also we will discuss around 20 MCQ questions. All these questions will be based on the week 6 course material of the medicinal chemistry, and all of the MCQ questions, it will have four options, and in the chat box I will ask for the answers for the every question, and you have to read the question carefully and look at the answers and let me know in the chat box which of the following will be the correct one. But before starting, let me just summarize few points. Like there will be this kind of problem solving session every week, and these sessions will be on Friday from 4 to 6 p.m. These sessions are live on Google Meet platform. This is the link for the every session, and this link will be same for our entire course. So you can use this link for joining this Friday class from 4 p.m. And now the important thing is, if there is any network disruption happens from my side, if there is some issue, so I will try to rejoin as soon as possible. Do not leave the session in between. Okay, I will try to rejoin as soon as possible. And if there is any network issue from your side, you can just rejoin at any time because this call is open, so there is no issue. During the sessions, the students can post their question, their doubts to the chat box. Since I'm recording the session, I have muted your microphones and also the camera so that I can record the session seamlessly. But the chat box is always open, and I am monitoring the chat box every now and then. So if there is any issue, any doubt, any problem, just let me know through the chat box. Okay. After the class, if the doubts persist, the students can drop an email to me. I will share my email ID at the end of the session, and also there is a discussion forum in the course page, so you can just go to the discussion forum and post your queries there too. And this presentation and this class recording, it will be shared to you after the session. Okay. So I will be starting the session. Just let me check one thing. Okay, so I hope my voice is audible and the screen is visible to you. If not, please kindly let me know. So as I have already mentioned, that what we will do in every live session, what we generally do is we discuss MCQ questions. Okay, I have designed this question in such a way that it will help you for to solve that particular week's assignment, and as well as it will help all the questions are discussed in a very elaborate manner. So if there is there are the subjective questions asked in the final examination, you can easily answer these questions after after going through all the class materials. Okay, along with this PPT and the course material is which is made available to you. I think it will be very helpful for you for the final examinations also. So today, as I have already mentioned, is our week 6 live session, and here we will be discussing the course content which are in the week 6 of this medicinal chemistry. So let us first check what are the course content of the week 6. So basically, week 6 course content are mainly of divided into the topics of pharmacokinetics. Okay, we will be discussing various aspects of pharmacokinetics, and also we will be discussing the drug metabolism, how the drugs are metabolized in our body. That topics which we will be discussing today. Okay. So I will be showing you the first question. Each question will have four options, and read the options very carefully and let me know in the chat box A, B, C or D which of the following will be the correct answer. Okay, and there is no marks for answering in the class. So if you, even if your answer is wrong, you don't have to worry about it. Okay, but there is but I want all of you to answer the question in the class so that I will understand that if you are understanding the course or not. Okay, so now let me start with the first question of today's session. So the first question of today's session, it says that which of the following statements best describes pharmacokinetics? Your options are: number A, the study of how drugs reach the target in the body and how the levels of a drug in the blood are affected by various factors. Number B, the study of how drugs can be designed using molecular modeling based on the drug's pharmacophores. Number C, the study of how the drug interacts with its target binding site at the molecular level. And number D, the study of which functional groups are important in binding a drug to its target site and the identification of the pharmacophore. So, which of the following statements best describes this word pharmacokinetics? Again, I will be repeating the options. Option number A says the study of how the drugs reach their target in the body and how the levels of a drug in the blood are affected by the various factors. Number B, the study of how drugs can be designed using molecular modeling based on the drug's pharmacophores. Number C, the study of how a drug interacts with its target binding site at the molecular level. And number D, the study of which functional groups are important in binding a drug to its target site and the identification of pharmacophore. So, which of the following statement is the correct description for this word pharmacokinetics? I will be give you some time. Read all the options very carefully and then let me know in the chat box A, B, C or D which of the following you think is the correct answer or the correct statement for describing pharmacokinetics. Which of the following statements best describes pharmacokinetics? Number A, the study of how the drugs reach their target in the body and how the levels of a drug in the blood are affected by various factors. Number B, the study of how drugs can be designed using molecular modeling based on the drug's pharmacokinetics. Number C, the study of how a drug interacts with its target binding site at molecular level. Number D, the study of which functional groups are important in the binding site of a drug for in the binding a drug to its target site and identification of the pharmacophore. One more minute and then I will check your answers in the chat box. And after that, we will discuss about pharmacokinetics in detail. Okay, let me check the chat box now. Okay, I have got answers which are saying that it is number A. Let me check if number A is the correct answer or not. So, the correct answer will be yes, it's number A, the study of how drugs reach their target in the body and how the levels of the drug in the blood are affected by various factors. So as it said by one of the students that ADME, yes, pharmacokinetics, it is also as ADME. So what is ADME or pharmacokinetics? So basically pharmacokinetics is the study of what our body is doing to a drug. Okay, so basically when we are taking a drug, how we are taking the drug, after taking the drug, where it is going, how it is getting metabolized, and how, what are the other things. So all these things are discussed in the pharmacokinetics. Okay. So, in the various aspects of the pharmacokinetics is basically the four different things are there in pharmacokinetics: absorption, distribution, metabolism, or excretion, which are commonly known as ADME. Okay, so A for absorption. So, absorptions mean the movement from the site of administration to the bloodstream. Okay, so the common routes of the drug administrations are the oral, intravenous, intramuscular, subcutaneous, and then transdermal and pulmonary or nasal. So there is the important factors or terms which are required in case of this absorption. One of the such term is the bioavailability, which is the extent or the rate at which the drug enters the systematic circulation. Bioavailability is 100% in case of the IV administration. Okay, so when there is the intravenous administration, the bioavailability of the drug is said to be 100. Okay, and there are other means of the administration also. The drug can be taken as in the orally, as in the form of the tablets, capsules, or any kind of your syrups and all. So it can be given by intravenous, by through injection, intramuscular way, transdermal, nasal drugs. Okay, and so these are the absorption. So absorption is that taking the drug in our body. Then the distribution. So what is the distribution? The transport through the systematic circulation to the target tissues and or organs. Okay, so it is the drug's journey from the systematic circulation to the target tissues of the organ. So it is known as the distribution. So what is so it is influenced by the adequate blood flow, ability of the drug to exit the vascular system, ability of the drug to enter the target cell. The factors that may limit the distribution is the vascular damage due to the chronic disease such as diabetes, limited blood supply to the abscesses or tumors, and the tight junctions of the blood-brain barrier. Okay, then comes the metabolism. So what is the metabolism? So chemical alterations or biotransformation of a drug to make it pharmacologically active or to facilitate the elimination. Okay, so primarily it occurs in liver, affects the metabolism, vary by drug, may include the conversion from lipid soluble or water soluble compound, inactivation of the drug, bioactivation of the prodrug, increase or decrease toxicity. And then this is the excretion. So most drugs are filtered by the kidneys and excreted through the body. So other routes are like through the our through the feces or through the urine, the drugs are getting excreted, or the sweats also. Okay, so these are the four aspects of the pharmacokinetics: absorption, distribution, metabolism, and excretion. So in short, the pharmacokinetics, what does it means that how our body is affecting a particular drug. Okay, so how our body is affecting a drug, it is known as the pharmacokinetics. So number A statement was the correct one. Okay, so now I will go to the next problem. So problem number 2 says, which organ is known as body's customs office? Your options are number A heart, number B brain, number C pancreas, and number D liver. So which of the following organ is known as body's customs office? Heart, brain, pancreas, or liver? Let me know in the chat box A, B, C, or D which of the following you think is the correct answer. Which organ is known to be the body's customs office? Heart, brain, pancreas, or liver? So let me check the chat box now. Okay, I have got only one answer which is saying it is number D. Others also try to answer the question. Even if your answer is wrong, it does not matter. But let me know what you think. Which organ is known to be body's customs office? Heart, brain, pancreas, or liver? Are there any other answer? Okay, so I will take liver as the answer. And okay. Let me now check which of the following will be the correct answer. So, body's customs office, the organ which is known to be body's office is very good. Number D, it is the liver. So let us understand what is or how a typical journey of an orally active drug happens. Okay, so the preferred method of the drug administration is the oral route, and so we shall consider some of the hurdles and the hazards faced by such a drug in order to reach its eventual target. When a drug is swallowed, it in the form, the drug may be swallowed in the form of the capsule, tablet, or in the liquid form. I have already mentioned it, right? So when a drug is swallowed, it enters the GI, which is the gastrointestinal tract, which comprises the mouth, throat, stomach, and the upper and lower intestine. A certain amount of the drug may be absorbed through the mucosal membrane of the mouth, but most passes down into the stomach, where it encounters the gastric juices and the hydrochloric acid. These chemicals aid in the digestion of food and will treat a drug in a similar fashion if it is susceptible to breakdown and it is not protected within the acid-resistant pill or capsule. For example, the first clinically useful penicillin was broken down in the stomach and had administered by injection. Other acid-labile drugs include the local anesthetics and the insulin. If the drug does survive the stomach, it enters to the upper intestine where it encounters digestive enzymes and serve to break down the food. Assuming the drug survives this attack, then it has to pass through the cell's lining of the gut wall. This means that it has to pass through the cell membrane of two occasions. First to enter the cell and then to the exit the other side. Once the drug is passed through the cells of the gut wall, it can enter the blood supply relatively easily as the cells lining of the blood vessels are loose fitting and there are pores through which most of the drugs can pass. In other words, drug enter the blood vessels by passing between the cells rather than through them. The drug is now transported in the blood to the body's customs office, the liver. The liver contains enzymes that are ready and waiting to intercept the foreign chemicals and modify them in such a way that they are mostly easily excreted, a process known as the drug metabolism. Following this, the drug has to be carried by the blood supply around the body to reach the eventual target, which may require crossing for the cell membranes, always assuming that it is neither excreted before it gets there nor diverted to the different parts of the body where it is not needed. It can be seen that the stringent demands are made on the orally administered drug. It must be stable in both chemical and enzymatic attack. It must also have the correct physiochemical properties to allow it to reach its target in the therapeutic concentrations. This includes the efficient absorption, effective distribution to the target tissues, and an acceptable rate of excretion. Okay. So this is basically a typical journey of an orally active drug when a drug is given to you. So the questions may be asked like this, like how this drug, what happens when you are taking an orally active drug and how it behaves in the body. So this is the points. Okay, you can write it in a point-wise manner also. So remember, liver is the most important organ in case of this drug metabolism because liver contains various enzymes and which will decide if which will intercept the foreign chemicals in our body to make it more labile for more excretion. Okay. So the correct answer was number D. Now let's go to the next problem. Problem number 3 says which of the following statements is true regarding the Lipinski's rule? Your options are number A, there are no exceptions to this rule. Number B, hydrogen bond donor and acceptor value must be equal to 5. Number C, cyclosporine does not obey this rule. Number D, this rule is qualitative and foolproof. So which of the following statements is true regarding the Lipinski's rule? Again, I will repeat the options. Your options are number A, there is no exceptions to this rule. Number B, hydrogen bond donor acceptor value must be equal to 5. Number C, cyclosporine does not obey this rule. Number D, this rule is qualitative and foolproof. Let me know in the chat box A, B, C or D which of the following statements is true. I have already mentioned in couple of times that whenever there is this kind of correct, incorrect, true, false type of questions, please read all the options very carefully. Most of the students they make mistake during this type of questions when this type of questions are asked because sometimes this can be a little confusing. So read all the options and let me know. Let me know what do you think. Okay, let's check the chat box. Okay, so I have got two answers. One is saying it is number B, another is saying it is number C. Okay, let me check which of the following is the correct answer. So, since I'm recording the sessions, I have muted your microphones and camera so that I can record it seamlessly. But if you have any doubt or any query, I have already said you, please let me know through the chat box. Okay. So I have got two answer for the problem number 3. One is saying that the statement number B, which says that hydrogen bond donor and acceptor value must be equal to 5, it is correct. And another one is saying that cyclosporine does not obey this rule. This statement is true. So now let me check which of the following will be the correct answer. So the correct answer will be, it is number C, which says the cyclosporine does not obey this rule. Okay, now let us see what is the Lipinski's rule. So basically, the Lipinski's rule of 5, it comprises of these rules. So okay, so this is the so there should not be more than 5 hydrogen bond donor, not more than 10 hydrogen bond acceptors. Molecular weight of a drug must be equal to 500, and C log P value does not greater than 5. Okay. So basically, the hydrophilic and hydrophobic character of a drug is the crucial factor affecting the absorption through the gut wall. In theory, the molecular weight of the drug should be irrelevant. For example, cyclosporine is successfully absorbed through the cell membranes, although it has molecular weight of 1200. Okay, in practice, however, larger molecules tend to be poorly absorbed. As a rule of thumb, orally absorbed drugs tend to obey the well-known Lipinski's rule of 5. Okay, the rule of 5 was derived from the analysis of compounds of the World Drugs Index database, aimed to identify features that were important in making the drug orally active. It was found that the factors concerned involved numbers that are multiples of the 5. Okay, molecular weight less than 500, no more than 5 hydrogen bond donor groups, no more than 10 hydrogen bond acceptor groups. A calculated log P value is less than 5, or the log P is a measure of the drug's hydrophobicity, which we will see later. The rule of 5 has been an extremely useful rule of thumb for many years, but it is neither qualitative nor foolproof. For example, orally active drugs such as the atorvastatin, the rovastatin, cyclosporine, the vinorelbine, they do not obey this rule of 5. It has been demonstrated that a high molecular weight does not itself cause poor oral viability. One of the reasons that the molecular weight appears to be important in that larger molecules are invariably have too many functional groups capable of forming hydrogen bonds. Another source of debate concerned that the calculation of the number of hydrogen bond acceptors. In Lipinski's original paper, the number of hydrogen bond acceptors corresponded to the total number of oxygen and nitrogen atom present in the structure. This was done for the simplicity's sake, but most of the medicinal chemists would discount the weak hydrogen bond acceptors such as the amide nitrogens. Therefore, it is better to view Lipinski's rule as a set guideline rather than the rules. Lipinski himself stated that the compound is likely to be orally active as long as it does not break one or more of his rules. Okay. So this is the structure of the cyclosporine, which has a molecular weight around 1200. Yet it is a very good orally active drug. Okay. So this was the Lipinski's rule of 5. It is very important to all of you to understand what is Lipinski's rule of 5. And the topic of this log P value or this things comes from the lipophilicity and the hydrophilicity. Okay. So this is depends on the partition coefficient. That's P for the partition coefficient and all these things which we will discuss later. Okay. So yeah, statement number C, it was true regarding this Lipinski's rule of 5. Now, let us go to the next problem. So problem number 4 says most of the current drugs are metabolized by dash primary CYP enzymes. Fill in the blanks. Your options are number 2, options number B is 7, option number C is 4, and option number D is 5. So most of the current drugs are metabolized by how many number of primary CYP enzymes? You have to answer me that, okay? Number 2, number 7, number 4, or number 5. What will be the correct number? Correct number of the primary CYP enzymes that will metabolize the most of the current drugs. I will give you 2 to 3 minutes, then I will check the chat box and then we will discuss that particular question. So what is the correct number of the primary CYP enzymes? Let me check the chat box. Okay. So I have got two answers. One is saying it is number B, another is saying it is number D. Okay, let me check which of the following will be the correct number. So, the most of the current drugs are metabolized by, it is number D, 5 primary CYP enzymes. Okay. So this is the relative importance of the CYP 450 drugs in the drug metabolism. So the major is the CYP3A family of enzymes. As you can see, it is taking the most of the place of this pie chart. Then we have the CYP2D6, then CYP2C, CYP1A2, and then CYP2E1. So basically, there are at least 33 different cytochrome P450, or in short CYP enzymes, grouped into four main families, CYP1 to CYP4. Then within each family, there are various number of subfamilies designed by a letter. Each enzyme within the subfamily is designated by the number. For example, CYP3A4 is the enzyme 4 of the subfamily A of the main family 3. Okay, most of the drugs are current use are metabolized by 5 primary CYP enzymes: CYP3A, CYP2D6, CYP2C9, CYP1A2, and CYP2E1. The isozyme CYP3A4 is particularly important in the drug metabolism and is responsible for the metabolism of the most drug. So this is very important. Okay, so remember this thing, that the isozyme CYP3A4 is particularly the most important in the drug metabolism and responsible for the metabolism of the most of the drugs. Okay, this one. So the correct answer was number 5, and I think there was a correct answer in the chat box too. So very good. So now I will go to the next problem. So problem number 5 says, which of the following, which metal ion is present in the active site of cytochrome P450? Your options are number A magnesium, number B nickel, number C zinc, and number D iron. So which of the following metal is present in the active site of CYT P450 or cytochrome P450? Let me know in the chat box, A, B, C, or D, which of the following you think is the correct answer. As I have already mentioned, there is no negative marking here. So even if your answer is wrong, it's fine. But please answer, since otherwise I won't understand that if you are following the topics of the course or not. Okay? I will give you some time. Think and let me know. So which metal ion is present in the active site of cytochrome P450? Manganese, nickel, zinc, or iron? Let me check the chat box. Okay, I have got two answers. Both are saying it is number D. Okay, let me check if it is the correct one or not. So, the metal ion which is present, so it is number D, iron. So, iron is the present in the active site of cytochrome P450. So basically, this is the structure of cytochrome P450. So we have not yet discussed what is the role or what is the major role of the cytochrome P450 this particular enzyme does. So the cytochrome P450 is involved in various biological systems or various works. Okay, and the main thing which cytochrome P450 is do is it is acts as the oxygenase. Okay, more monooxygenase. So basically, it inserts the oxygen into the system or a particular bond. So as for example, as you can see here, that there is a normal sulfur hydrogen bond, and this particular enzyme, what it will do, it will insert an oxygen, and this oxygen is taken from the atmospheric oxygen. Okay, so this is a normal oxygen molecule. This oxygen atom comes from. So this is the active site, and there, as you can see, there is a iron 3 here in the active site. Okay, and it is a porphyrin ring, and we have a cysteine residue here in the this position, and then there is a this NADPH is also there in presence of oxygen, and it will form this kind of structure, and there will be electron pull, and this is the active site. This is the first the active species, which is the iron 4 cation radical. Okay, so as you can see, there is a cation plus there is a radical. Here iron is in the 4 oxidation state, and then this is the complete reaction, how it works. Okay, this thing is not in your syllabus, but I have just only given it for your understanding. And the cytochrome P450 is involved in various metabolisms or various works like xenobiotic metabolism. Okay, cytochrome CYP8A1, it metabolizes several herbicides. There's hormone regulation, CYP707ABA8 prime hydroxylase, the ABA catabolism. Secondary metabolite biosynthesis, CYP7A79A is the urine biosynthesis. Then plant defense, antioxidant biosynthesis, fatty acids metabolism, and so on. The cytochrome P450 is a very important enzyme in our body, which is involved in various mechanisms. Okay, and iron is present in its active site. Okay? So now I will go to the next problem. Problem number 6 is, which of the following equations is the correct for the Henderson-Hasselbalch equation for weak bases? Your options are: number A, pH equal to pKa minus log unionized drug concentration by ionized drug concentration. Number B, pH equal to pKa minus log of the ionized drug concentration divided by unionized drug concentration. Number C, pH equal to pKa plus log of unionized drug concentration and ionized drug concentration. Number D, pH equal to pKa plus log of the ionized drug concentration and unionized drug concentration. So what will be the correct term for the correct Henderson-Hasselbalch equation for the weak bases? This equation is very important in case of acid base, and the equation is little different for the acid and bases. So here you have to identify what will be the correct Henderson-Hasselbalch equation for the weak base. Option number A, pH equal to pK minus log of ionized drug concentration by ionized drug concentration. Number B, pH equal to pK minus log of ionized drug concentration by unionized drug concentration. Number C, pH equal to pK plus log of unionized drug concentration by ionized drug concentration. And number D, pH equal to pK plus log of ionized drug concentration by unionized drug concentration. So What will be the correct equation? Okay, let me check the chat box now. I got two answers. One is saying it is number C, another is saying it is the number D. Okay, let me check which of the following will be the correct answer. So, the correct answer will be, it is number C. pH equal to pK plus log of unionized drug concentration by ionized drug concentration. So remember, the question was asked for the weak basis. Okay. In case of acid, the equation will be a little different. So which I will show you later. So basically, the extent of the ionization of a particular pH can be determined by the Henderson-Hasselbalch equation, which says it is the pH equal to pK plus log of RNH2 by RNH3 plus. Okay, so this is for the base. Okay, this is for the weak base. This equation is given here. Okay, so the RNH2 is the concentration of the free base, and the RNH3 plus is the concentration of the ionized amine. K is the equilibrium constant for the equilibrium shown here. Okay, so this is the equilibrium, and the Henderson-Hasselbalch equation can be derived from the equilibrium constant. So what is the equilibrium? So this is between this. This is the ion receptor interaction with the water solubility, and this is the ionized imine, and this is the non-ionized amine. So K equal to H plus into the concentration of H plus into the concentration of RNH2 divided by the RNH3 plus. Therefore, K equal to minus log of H plus RNH2 divided by RNH3 minus. So if you take this is just separated this out, and this equation will be like this: pH equal to pK plus log of RNH2 divided by RNH3 plus. So note, when the concentration of the ionized and unionized amine are identical, the ratio will be 1 and log 1 equal to 1. So Henderson-Hasselbalch equation will be simplified to pH equal to pK. In other words, the amine is 50% ionized at the pH equal to pK. Therefore, drugs of the with a pK of 6 to 8 are approximately 50% ionized at the blood pH or slightly acidic pH in the intestine. Okay, so this is the simple derivation how it comes. Okay, so this was the Henderson equation for the weak bases. Okay, so now I will go to the next problem. Problem number 7 says pK of the most acidic functional group in the antibiotic cefotaximine is 3.4. So now predict where it will be absorbed in the body. So number A, stomach having the pH of 1. Number B, intestine having the pH of 6. Number C, in both. Number D, in none. So pK is the move of the most acidic functional group. So now here the term comes for the acidic group. Okay. In the previous one we were discussing regarding the base, but now here we are discussing about the acids. So the pK of the most acidic functional group of the antibiotic cefotaximine is 3.4. So predict where it will be absorbed in the body. The stomach having the pH of 1 or intestine having the pH of 6. Number C, in both. Number D, in none. So where it will be absorbed in the body? So now let me check the chat box. Okay, so I have got a few answers, I think. Number B, number A, and number A. So I have got one number B and two number A. Okay, let me check which of the following will be the correct answer. The correct answer will be yes, it is number A, stomach having the pH of 1. Now I have already mentioned you in the previous question we were discussing about the base. The thing is same if you write the equation and solve it. So you will find that in case of acids, the equation is like this: pH equal to pK plus log of ionized form divided by unionized form. Okay. So in stomach, the pH is mentioned to be 1 and the pK of this group, okay, this functional group is 3.4. If you substitute this thing, so you will find that 1 equal to 3.4 plus log of carboxylate ion divided by carboxylic acid, or the carboxylate ion divided by carboxylic acid will be antilog of 1 by 3.4, which will be 0.00398, which indicates the drug will remain as a whole rather than the unionized form, rather than ionized form. Okay. Because this concentration of this carboxylic acid is greater, then in the intestine, what is in the intestine happens? The pH is 6 equal to 3.4 plus this on. So the carboxylate ion divided by carboxylic acid is 2.6, which is greater than 1, which indicates that drug will be mainly in the carboxylate ion. Okay, so unionized form of the drug can get a better access to the membrane, so it will be get absorbed in stomach. Okay, so since the drug is in the most unionized form in the stomach, okay, and this unionized form of a drug can get a better access to the membrane, that's why this particular drug will be absorbed in the stomach where the pH is 1, because in the stomach where the pH is 1, the drug will remain as a whole rather than in the ionized form. Okay, so number A was the correct answer. So now I will go to the next problem. So problem number 8 says the process where the drug is engulfed by the cell membrane and a membrane bound vesicle carry the drug across the cell is known as, your options are number A osmosis, number B diffusion, number C pinocytosis, number D mitosis. So what is that process known where the drug is engulfed by the cell membrane and a membrane bound vesicle carry the drug across the cell? Osmosis, diffusion, pinocytosis, or mitosis? All of you, please try to answer the questions. Even if your answer is wrong, it is completely fine, but please try to answer the question. Identify the process where the drug is engulfed by the cell membrane and a membrane-bound vesicle carry the drug across the cell. Or in the process, what is this process known? Osmosis, diffusion, pinocytosis, or mitosis? The process where the drug is engulfed by the cell membrane and the membrane-bound vesicle is formed which carry the drug across the cell. What is this process known? Let me check the chat box. Okay. So I have got three answers and all the three are saying it is the number C. Okay, let me check if number C is the correct answer. So the correct answer will be very good. It is number C, pinocytosis. So occasionally, polar drugs with a high molecular weight can cross the cells of the gut wall without actually passing through the membrane. This involves a process known as pinocytosis, where the drug is engulfed by the cell membrane and a membrane-bound vesicle is pinched off to carry the drug across the cell. This vesicle then fuses with the membrane to release the drug with the other side of the wall. So there is a drug, then it is making the vesicle, and the vesicle is fusing to the other side. So this process is known as the pinocytosis. So what are the steps? So first, the solutes bind to the cell membrane. This is the solutes of the drug. This is the cell membrane. This is the nucleus, and this is the cytoplasm. Okay. Then now what happens? The cell membrane start to fold inward like this, and then the cell membrane continues to fold, and the both ends of the folds are met, and the producing a pinosome. So this vesicle is also known as the pinosome. Okay, and the process is known as the pinocytosis. Now, this pinosome will go to the other side and it will fuse with the cell membrane to release the solutes of the compound. So, this is the known as this process known as the pinocytosis. Okay. So, now I will go to the next problem. So, problem number 9 says, which of the following statements is the closest description of the phase 1 metabolism? Your options are number A, reactions which occur in blood supply, number B, reactions which add a polar functional group to drug, number C, reactions which occur in the gut wall, and number D, all of these. Which of the following statement is the closest description of the phase 1 metabolism? Your options are number A, reactions that occur in the blood supply, number B, reactions which add a polar functional group to the drug, number C, reactions which occur in the gut wall, and number D, all of these. Which of the following statements will be the closest description of the phase 1 metabolism, A, B, C or D? Number A, number B, number C, or all of the now A, B, C are the correct? Let me know in the chat box. Let me check if there are any answers for the problem number 9. Okay, so I have got few answers. I think I have got only two answers. Both are saying it is the number B. Let me check if it is the correct one or not. So, the statement which is the closest description of the phase 1 metabolism will be, yes, it is the number B, which says that the reactions which add a polar functional group to a drug. Okay, so in phase 1 metabolism, basically this happens, the reactions which add a polar functional group to a drug. This all the reactions are termed in the case of the phase 1 metabolism. So in the phases, what are the phases of metabolism? Phase 1 and phase 2 are the two major phases of the metabolism. In the first one, the functionalization reactions happen, which converts the parent drug to the more polar metabolite by introducing or unmasking a functional group like OH, NH2 or SH. And phase 2 are the conjugation reactions, the subsequent reaction in which the covalent linkage is formed between the functional group and the parent compound or the phase 1 metabolite and endogenous substrates such as the glucuronic acid, sulfate, acetate or an amino acid. So phase 1 and phase 2 metabolisms overview. Phase 1 reactions are often make a drug sufficiently polar and it can be excreted. However, phase 2 reactions are often required whereby a conjugation is added or covalently linked such as glucuronic sulfuric acetic acid to make the drug even more polar charged or hydrophilic. Conjugations are often occurs at the functional group location. So parent drug phase 1 reaction from this parent drug to the functional group and add a functional group to polar functional group here, and the phase 2 it conjugates this polar functional group to another moiety. Okay, although phase 1 reactions usually occur before the phase 2 reaction, sometimes the order is reversed. The phase 2 reactions can occur alone also. So parent drug, first the phase 1 reactions happen. So more polar metabolite is pharmacologically uncharged, inactive or enhanced or otherwise modified. And this after phase 1 reaction directly it can go to the elimination, or phase 1 reaction can go to the phase 2 reaction where the addition of conjugate happens, also go to the elimination. All this metabolism, the phase 1 and phase 2, all this makes this parent drug more which is the mostly lipophilic, to more hydrophilic so that it can be excreted easily. Okay, so this is the overview for the your phase 1 and phase 2 metabolisms. Okay, so now I will go to the next problem. So problem number 10 says, which of the following conversions is most unlikely to happen in the presence of cytochrome P450? Your options are number A, RCHO to RCH2OH, number B, RCH to CH3 to RCOCH3, number C, RR-CHX to RCOR-, and number C, ARCH2R to ARCHOHR. Which of the following conversions are most unlikely to happen in presence of cytochrome P450? Number A is the conversion of aldehyde to alcohol. Number B is the conversion of alkene to ketone. Number C is the conversion of your halite, haloalkene to the ketone. Or number D is the conversion of an alkene to the alcohol, but with the aromatic functional group. Which of the following conversions is most unlikely to happen in presence of cytochrome P450? Think what cytochrome P450 does and then answer the question. I will give you some time, 2 to 3 minutes time, and then I will check the chat box. Let me know A, B, C or D, which of the following you think is the correct answer for problem number 10. Let me check the chat box. Okay, I think I have got quite answers. Number A, number C. So I have got only two answers. One is number C and one is number A. Okay, let me check which of the following is the correct answer. So the correct answer will be, it is number A, conversion of the aldehyde to alcohol. It is the most unlikely to happen in presence of cytochrome P450. Now first understand what does this cytochrome P450 do. I have already mentioned it, I think, in our previous slides, that the enzymes that constitute the cytochrome P450 family are most important metabolic enzymes and are located in the liver cells. They are the hemoproteins containing the heme and iron, and they catalyze a reaction that splits the molecular oxygen such that one of the oxygen atoms is introduced into the drug and the other ends up into the water. As a result, they belong to a general class of enzymes called the monooxygenase. So the drug, in presence of oxygen, NADPH and proton, it constitutes the drug with it adds the oxygen to the drug, and it converts the NADPH to NADP and forms the water. Okay, so oxidation reactions are basically happens in presence of the cytochrome P450 enzymes. Okay. So now, what are the which oxidative reactions are catalyzed by cytochrome P450? So as if you just look at the options, the first reaction itself, it is a reduction reaction. Okay, the formation of the aldehyde to alcohol. This conversion is a reductive reduction. Basically, reduction happens here. So this is the most unlikely to happen in presence of cytochrome P450. So what are the reaction that is catalyzed by these particular enzymes? So oxidation of the terminal methyl groups like this RaCH3 to RCH2OH to RCHO to RCOOH, and this terminal alkene group can be oxidized by this enzyme. Also, the oxidation of the penultimate carbon on the alkyl substituent. Okay, so the penultimate means the this carbon atom, which is the second last from here, which also can get oxidized in presence of the cytochrome P450 enzyme. Okay, so oxidation of the penultimate carbon on alkyl substituents. The oxidation of the most exposed or the most activated region of the alkyl ring. This reaction can also happen in presence of cytochrome P450. Then oxidation of the activated carbon centers next to the sp or sp2 carbon, this like this. Okay, within presence of aromatic carbons. Okay, or this also can be done with this particular cytochrome P450. The dealkylation of amine ethers and thioethers via oxidation of the activated carbon, including the demethylation, can also happen like this. Okay. These reactions can be also catalyzed by cytochrome P450, and dehalogenation reactions of the alkyl halides are also facilitated by this cytochrome P450. Okay, if there is a halogen, first it is converted to like this. First it is hydroxylated, and then this halogen is eliminated and we get the ketone. So this dehalogenation reaction can also be catalyzed by the cytochrome P450 enzyme. Okay, so this all the reactions are catalyzed by this family of enzymes. Okay, and all these transformations all these transformations fall into the category of phase 1 metabolism. This is also important thing which you have to remember. Okay, so this was the problem number 10. Now I go to the next problem. Problem number 11 says alkenes and aromatic groups can be metabolized to diols. Which enzymes are involved in this case? Number A, cytochrome P450 enzyme. Number B, epoxide hydrolase. Number C, both of the above. Number D, none of the above. Alkenes and aromatic groups can be metabolized to diols. Which enzymes are involved in this conversion? Number A, cytochrome P450. Number B, epoxide hydrolase. Number C, both of the above. Number D, none of the above. Alkenes and aromatic groups can be metabolized to diols. Which enzymes are involved here? Let me know in the chat box. Okay, let me check the chat box now. So, what will be the correct answer for problem number 11? Okay, I have got three option answers, and all the three are saying it is number B. Okay, let me check if number B is the correct answer or it is or not. So the correct answer will be number C, both of the above. Okay, so both of the above reactions can be done by this particular enzymes like the cytochrome P450 and epoxide hydrolase. Okay, cytochrome P450 I have already mentioned that it can convert the alkenes and aromatic groups to diols. Okay, I have not mentioned it directly, but it can also be happen in presence of the cytochrome P450. Okay, just let me highlight this. So cytochrome P450 enzymes can catalyze the oxidation of unsaturated sp2 and sp carbon centers present in the alkene, alkyne and aromatic ring. In case of alkene, a reactive epoxide is formed and which is after deactivated by enzyme epoxide hydrolase to form the diol. Okay, in some cases the epoxide may evade the enzyme. In this, if it happens, then what happens? In the epoxide, if it happens, it can act as an alkylating agent and react with the nucleophilic group present in the proteins and nucleic acid, leading to toxicity. The oxidation of an aromatic ring results in a similarly reactive epoxide intermediate, which can have several possible fate. It may undergo rearrangement reaction involving a hydride transfer to form the phenol, normally the para position. Alternatively, it may be deactivated by the epoxide hydrolase to form a diol and the react with the glutathione S transferase to form a conjugate. If epoxide intermediate evades these enzymes, it may act as a alkylating agent to prove toxic. Electron-rich aromatic ring are likely to be epoxidized more quickly than with the electron withdrawing substituent. That has consequences for the drug design. Okay, so basically what happens, this alkene, this oxygen, in case of alkene and unsaturated aromatic unsaturation, wherever there is a unsaturation, this oxygen insertion will always be done by the cytochrome P450 enzymes. Okay, and after that this conversion of this epoxide to this diol, it will be performed by the epoxide hydrolase. Okay, so these two, both of the enzymes, this epoxide hydrolase and also the cytochrome P450 is the responsible for the for catalyzing the or for the converting the alkene and aromatic group to diols. Okay, first oxygen is added and epoxide is formed with cytochrome P450 and then epoxide hydrolase. What does it do? It hydrolyzes the epoxide and forms this diol. That's how it happens. Okay, and the very important thing is whenever there is a there is this aromatic ring like benzene ring, so this rearrangement will occur and we will get usually a para position hydroxyl rather than the in case by a hydride shift or in presence of epoxy hydrolase we can get a diol like this. Okay, so yes, epoxide hydrolysis number B was correct answer, but cytochrome P450 that acts first. Okay, after that epoxide hydrolase forms the diol. So both of the above will be the correct set of enzymes. Now I will go to the next problem. So problem number 12 says identify the incorrect statement. Your options are number A, cytochrome P450 can convert amine to N oxide. Number B, cytochrome P450 cannot oxidize heteroatoms. Number C, cytochrome P450 can convert thiol to disulfide. Number D, cytochrome P450 can perform desulfurization. Desulfurization. So, what are the options? Which of the following is the incorrect statement? Number A, cytochrome P450 can convert amine to N oxide. Number B, cytochrome P450 cannot oxidize heteroatoms. Number C, it can convert thiol to disulfide. Number D, it can perform desulfurization. Identify the incorrect statement. Let me know A, B, C or D, which of the following you think is the incorrect statement? So let me check the chat box now. Okay, I have got answers like number D, and number C. So I have got two number D and one number C. Okay, let me check which of the following will be the correct answer. So the incorrect statement will be number B. Cytochrome P450 cannot oxidize heteroatoms. This statement is incorrect. Okay. So, tertiary amines are oxidized to N oxide as long as the alkyl group are not sterically demanding. Primary and secondary amines are also oxidized to the N oxide because they are rapidly converted to hydroxylamine and beyond. Aromatic and primary amines are also oxidized in the stages of aromatic nitro group, a process which is related to the toxicity of aromatic amine, as highly electrophilic intermediates are formed which can alkylate the proteins and nucleic acid. Aromatic primary amines can also be methylated in the phase 2 reaction to the secondary amine, which can undergo phase 1 oxidation to produce formaldehyde and primary hydroxylamines. Primary secondary amides can be oxidized to hydroxylamine, the functional groups that may link to toxicity and carcinogenicity. The thiols can be oxidized to disulfide. There are evidence that thiol can be methylated and methyl sulfide, and which are oxidized to sulfides. Okay, so these all reactions which are which can be which can be done by the cytochrome P450 enzyme. So amine can be converted to N oxide. As you can see here, all the transformations of amine to N oxides. Also, amines and aromatic amines can be converted to nitro group. Then we have this the aromatic heterocycles, which is like pyridine, these are also converted to the N oxide. So this is oxidation of nitrogen containing functional group. We have this oxygen. This particular cytochrome P450, it can oxidize the heteroatoms, as we can clearly see from these examples. Then thiol can be converted to disulfide. Okay, the thiol can be converted to methyl sulfide, to sulfoxide, to sulfone. Also, oxidation of sulfur containing group can also happen, and desulfurization also can happen where this sulfur C double bond S, which is this kind of this can be converted to this ketone group. Okay, the sulfur can be eliminated and can be replaced by the oxygen. And then we have this R3P, which is the phosphorus, which is also can be converted to the oxides in presence of the cytochrome P450. So all these conversions can be done by the cytochrome P450. So the statement which says that number B, which says that CYP450 cannot oxidize heteroatom, it is incorrect. Okay. So now I will go to the next problem. Problem number 13 says indinavir is metabolized by dash. Your options are number A, glutathione transferase, number B, cytochrome P3A, number C, epoxide hydrolase, and number D, CYP2D1, 2D9. Which of the following enzyme metabolizes this indinavir drug? Number A, glutathione transferase, number B, CYP3A, number C, epoxide hydrolase, and number D, CYP2D9. Let me know in the chat box A, B, C, or D which of the following will be the correct answer. Indinavir is metabolized by which enzyme? Glutathione transferase, CYP3A, epoxide hydrolase, CYP2D9. Okay, let me check the chat box. I have got only one answer, which is said is number B. Okay, let me check if number B is the correct answer or it is not. So the correct answer will be yes, it is number B, CYP3A. This enzyme metabolizes the isindinavir. So this is the structure of indinavir. As you can see, the structure is a little bit complicated. So basically, indinavir is an antiviral agent used in the treatment of HIV and is prone to metabolism, resulting in 7 different metabolites. Studies have shown that CYP3A subfamily of cytochrome P450 enzymes is responsible for 6 of these metabolites. These metabolites concerned arise from the N-dealkylation of the piperazine ring. Okay, so this is the piperazine ring, and this is this here. This N-dealkylation happens. So the N-dealkylation of piperazine ring, the N-oxidation of the pyridine ring. This pyridine ring N-oxidation can happen. Okay, then para-hydroxylation of the phenyl ring. Okay, this phenyl ring para-hydroxylation can happen in this position, and hydroxylation of the Indian ring. This is the Indian ring. Hydroxylation can happen here also. The seventh metabolite is the glucuronide conjugate of the pyridine ring. Okay, the pyridine ring, apart from N oxide, glucuronide also can be formed here. All these reactions occur individually to produce 5 separate metabolites. The meaning 2 more metabolites arise from the 2 more metabolic reaction taking place in the same molecule. Okay. So the major metabolites are these resulting from the dealkylation. As a result, the research has been carried out to try to design indinavir analogs that are resistant to this reaction. For example, structures having 2 methyl substituent on the activated carbon here in this position next to the pyridine ring has effective in blocking the dealkylation. Okay, so the dealkylation which happens in general. So if this activated position is blocked by the 2 methyl group, so what will happen? This dealkylation can be blocked. Okay, so if the drug is metabolized too quickly, so what will happen? This drug will not get able to hit the target. So sometimes we need to slower the metabolism of a drug by blocking a few step here or there. Okay. So this is the metabolism of indinavir, and the enzyme which is responsible, or the subfamily which is responsible is the CYP3A. Okay. Now I will go to the next problem. Problem number 14 says, which of the following enzymes are involved in the phase 1 metabolism? Your options are number A, flavin monooxygenase, number B, aldehyde dehydrogenase, number C, monoamine oxidase, number D, all of these. Which of the following enzymes is involved in phase 1 metabolism? Your options are number A, flavin monooxygenase, number B, aldehyde dehydrogenase, number C, monoamine oxidases, number D, all of these. Let me know in the chat box A, B, C or D which of the following will be the correct answer for problem number 14. Which of the following enzymes is involved in the phase 1 metabolism? Let me check the chat box. I have got one answer which is saying it is number D. Others are not answering. Okay, let me show what is the correct answer and let us discuss about it. So the enzyme which is involved in the phase 1 metabolism will be, yes, it is number D. Apart from CYP450, flavin monooxygenase, aldehyde dehydrogenase and monoamine oxidase, all these enzymes are involved in the phase 1 metabolism. Okay. So basically, phase 1 reactions I have already mentioned is the addition of a polar functional group to the drug. Okay. So what are the reactions what are catalyzed by flavin monooxygenase? So as the name suggests, monooxygenase means it will add the one oxygen. Okay. So as you can see, the similar reaction it can perform like our cytochrome P450. It can perform the N-oxide formation or the oxidation of heteroatoms, also the formation of disulfide, the hydroxylation of the amines, also the formation of the oxidation of the sulfur containing groups. It can be done by the flavin monooxygenase, alcohol dehydrogenase, okay, and the aldehyde dehydrogenase. What does this dehydrogenase mean? It will remove the hydrogen, mean it will perform the oxidation reaction. Okay, so the primary from primary alcohol to aldehyde, aldehyde to carboxylic acid, all these reactions are performed with the enzyme dehydrogenase. It can be alcohol dehydrogenase one when it oxidizes the alcohol to aldehyde, and aldehyde dehydrogenase when it oxidizes the aldehyde to carboxylic acid. Also we have the monoamine oxidase. What does this monoamine oxidases do? They convert the amine to aldehyde. Okay, the RCH2NH2, it converts this CH2NH2 to CHO. So these are the phase 1 oxidative reaction which is catalyzed by the miscellaneous enzymes, and the enzymes are the flavin monooxygenase, dehydrogenases, alcohol dehydrogenase and aldehyde dehydrogenases, and we have this monoamine oxidases. Okay, so all these reactions are involved in the phase 1 metabolism. Now I will go to the next problem. So problem number 15 says selegiline is a specific inhibitor of, your options are number A myo B, number B myo A, number C acetylcholine and number D ACE. Selegiline is a specific inhibitor of which enzyme? Myo B, myo A, acetylcholine esterase, or your ACE, which is the angiotensin converting enzyme. Let me know in the chat box which will be the correct answer for problem number 15. Selegiline is a specific inhibitor of which enzyme? Selegiline is a specific inhibitor of which enzyme? Monoamine oxygenase, B, or myo A, or acetylcholine esterase, or ACE. Let me check the chat box. I've got two answers. Both are saying it is the number A. Let me check if number A is the correct answer or not. So the correct answer will be yes, it is number A, MyoB. So monoamine oxidase inhibitors or MAOIs are the powerful class of antidepressant drugs. Okay, so these are very particularly effective in the treating atypical depression, panic disorder, or social phobia. Due to potentially lethal dietary and drug interaction, MAOIs are reserved for the last line of treatment, used only when other classes of antidepressant drugs like SSRIs and TCAs have failed. So basically, monoamine oxidase inhibitors. There are two isotypes of this enzyme. One is the A, MyoA, and another is MyoB. So present in the cytoplasm, MyoA is present in the cytoplasm and the neuron, CNS, and the peripheral tissues like liver. It acts non-specifically on Na, 5-HT, and dopamine. Chlorgyrin is a specific inhibitor of MyoA, and MyoB present mainly in CNS and acts more on dopamine. Selegirin is a specific inhibitor of MyoB, and paraglide is a non-selective MyoI. Okay. So monoamine oxidase, these are the very crucial antidepressant drugs. Okay, and monoamine oxidases I have already mentioned earlier also. So these are this reaction which is it takes places parts is the like in phase 1 metabolism it happens. Okay, and this two types of in the two isotypes, the examples are very important. Like chlorgyrin is the specific inhibitor of MyoA, whereas the selegirin is the specific inhibitor of MyoB, and parglylin is a non-selective MyoI. Okay, neither it targets specifically MyoA or nor MyoB. Okay. So this was the problem number 15. Now I will go to the next problem. Problem number 16 says phase 2 metabolism consists of dash reaction. Your options are number A, hydrogenation, number B, reduction, number C, conjugation, number D, cleavage reaction. So phase 2 metabolism consists of which type of reactions? Hydrogenation, reduction, conjugation, or cleavage? A, B, C, or D? Let me know in the chat box. Phase 2 metabolism consists of dash reactions: hydrogenation, reduction, conjugation, or cleavage. So phase 2 metabolism consists of which type of reactions? Already I think I have mentioned it a little bit. Let me check the chat box. Okay. Number C is the given answer. Let me check if number C is the correct answer or not. So, phase 2 metabolism, it consists of, yes, number C, conjugation reactions. So most of the phase 2 reactions are the conjugation reaction catalyzed by the transferase enzymes. The resulting conjugates are usually inactive, but there are exceptions to this rule also. So these are the glucuronidation of miscellaneous functional group. Okay, so basically this glucuronidation, it happens like of the alcohol, phenol, and carboxylic acid majorly. So this is the UDFP glucuronidate. Okay, and this is the glucuronyl transferase enzyme which makes this conjugate here, and these are the miscellaneous functional group where this particular conjugation can form. Okay, amides, amine, hydroxylamine, tertiary amine, sulfonamide, thiol, all these functional group, in all this functional group, this glucuronidation can happen with the help of UDFP, which transfers this group to this functional groups. Okay. Then we have the formation of the amino acid conjugates also, like in the drug, in presence of ATP and acyl-CoA transferase. First, the AMP conjugate is formed. Then we have this coenzyme A, which forms this CoA, and then amino acid and amino acid N transferase can transfer the amino acid to by forming this type of conjugates. Then we have this different type of conjugation reaction. We have the formation of the glutathione and mercury acid conjugates of the alkyl halides. Okay, like alkyl halide, this is the glutathione, and in the presence of glutathione transferase, this glutathione conjugate can be formed, and peptidases can form the cysteine residue here. And also we have this mercury acid conjugate, and also formation of the glutathione conjugates of the electrophilic groups. Okay, and this can also happen within presence of epoxide. The epoxide can also form a hydroxyl and the glutathione conjugate, and also here in presence of the alkenes and in the presence of this sulfones, this kind of conjugate can happen. So these are the various examples of the phase 2 metabolism conjugation reaction, where different type of conjugation and group transfer reactions happen. Okay. Now I will go to the next problem. Problem number 17 says sulfation is one important method of the detoxification of xenobiotics. Which of the following acts as the source of sulfate? Your options are number A, hydrogen sulfide, number B, methionine, number C, sulfuric acid, number D, 3-phosphoadenosine 5' phosphosulfate. Again, I will repeat. Sulfation is an important method of the detoxification of xenobiotics. Okay, which of the following acts as a source of sulfate? Your options are number A, hydrogen sulfide, number B, methionine, number C, sulfuric acid, number D, 3-phosphoadenosine 5' phosphosulfate. Which of the following will be act as a source of sulfate in the sulfation reactions? Let me know in the chat box, A, B, C or D, which of the following you think is the correct answer? Let me check the chat box. Okay, I have got only one answer, which is saying it is number D. Okay, let me check if number D is the correct answer or not. So, the correct answer will be, yes, it is number D, which is the phosphoadenosine or 3' phosphoadenosine 5' pyrophosphate. Phosphosulfate, sorry. So this is the structure of this 3' phosphoadenosine 5' phosphosulfate. Okay. So another form of the conjugation is the sulfate conjugation. This is the less common than glucuronidation and restricted mainly to phenol, alcohol, arylamine and N-hydroxy compounds. The reaction catalyzed by the sulfotransferases using the cofactor 3'-phosphoadenosine 5'-phosphosulfate as a sulfate source. Primary and secondary amines, secondary alcohol, phenol from stable conjugates, whereas primary alcohols from reactive sulfates, which can act as a toxic alkylating agent. Aromatic hydroxylamine and hydroxylamides also from unstable sulfate conjugates that can be toxic. So this is the sulfotransferases. So what does the sulfotransferases do? They, in presence of this 3'-phosphoadenosine 5'-phosphosulfate as a sulfur source, they form the sulfur conjugates from the alcohols and amines. So this is the example of the sulfur conjugation phase 2 reactions. Okay. This is a very important, and this is the structure of this particular phosphoadenosine phosphosulfate. Okay. Now I will go to the next problem. Problem number 18 says which of the following statements is incorrect regarding the blood-brain barrier? Your options are number A, the walls of the capillaries supplying blood to the brain are such which makes it difficult for polar groups to leave the capillaries. Number B, the capillaries in the brain have a fatty coating making it more difficult for polar drugs to enter the brain. Number C, the inner walls of capillaries supplying the blood to the brain are hydrophobic in nature. Number D, hydrophilic drugs pass through the blood-brain barrier more easily than the hydrophobic drugs. Which of the following statements is incorrect regarding the blood-brain barrier? Again, I will repeat the options. Number A, it says that the walls of the capillaries carrying blood to the brain are such that which makes it difficult for the polar drugs to leave the capillaries. Number B, the capillaries of the brain have a fatty coating making it difficult for the polar drugs to enter the brain. Number C, the inner walls of capillaries supplying blood to the brain are hydrophobic in nature. And number D, hydrophilic drugs pass through the blood-brain barrier more easily than the hydrophobic drugs. Which of the following statements is incorrect regarding this blood-brain barrier? Read all the options very carefully and then let me know in the chat box which of the following statement you think is incorrect regarding the blood-brain barrier. I will give you 2 to 3 minutes, then I will check the chat box and show you, and we will discuss this question. Which of the following statements is incorrect regarding the blood-brain barrier? Let me check the chat box now. I have got two answers. Both are saying number D is the correct one. Means number D is the incorrect statement regarding blood-brain barrier. Let me check if it is correct or not. So the statement which is incorrect regarding the blood-brain barrier will be, yes, it is number D, which says that hydrophilic drugs pass through blood-brain barrier more easily than the hydrophobic ones. So what is blood-brain barrier or BBB? We can also say it as BBB. So it is formed by a single layer of endothelial cells that line the inner surfaces of the capillaries of the brain. Okay. The semi-permeable capillary membrane, that is, it allows some materials to cross but prevents others from crossing. In most parts of the body, the capillaries are lined with the endothelial cells. The endothelial tissue has small spaces between each individual cell, so substances can move readily between the inside and outside the vessel. However, in the brain, the endothelial cells fit tightly together and substances cannot pass through the bloodstream. Some of the molecules, such as glucose, are transported out of the blood by the special method like active transport. Physiology: in the brain, endothelial cells are packed much tighter together due to the existence of the zonular occludence, the tight junction between them, blocking the passage of most of the molecules. The blood-brain barrier blocks all the molecules except those which cross the cell membrane by means of the lipid solubility, such as the oxygen, carbon dioxide, ethanol, steroid hormone, and all these are allowed by specific transport systems such as the sugars, some amino acids, substances with the molecular weight rather than 500 Daltons and generally cannot cross. Okay, so the statements, the first one which says that walls of capillaries supplying blood to the veins, which makes it difficult to polar drugs to leave the capillaries, is correct. The capillaries in the brain have fatty coating, making it difficult for the polar drugs to enter. It is also correct. The inner walls of capillaries supplying the blood into the brain are hydrophobic in nature is also correct. The hydrophilic drugs can pass through the barrier than the hydrophobic drugs. It is not correct. Okay, the opposite will be the correct one. So this is all about the blood-brain barrier. Now, next question, problem number 19 says low concentration of neurotransmitter dopamine has been implicated in the Parkinson disease and L-DOPA is the most widely used medicine to slow down the disease. Which of the following statements is incorrect? Your options are: number A, dopamine cannot cross blood-brain barrier. Number B, L-DOPA acts as a prodrug. Number C, L-DOPA can cross blood-brain barrier. And number D, L-DOPA is the first decarboxylated by the dopamine decarboxylase before entering the brain. Which of the following statements is incorrect regarding the L-DOPA? Your options are: number A, dopamine cannot cross the blood-brain barrier. Number B, L-DOPA acts as a prodrug. Number C, L-DOPA can cross blood-brain barrier. And number D, L-DOPA first decarboxylated by the dopamine carboxylase before entering the brain. Which of the following statements is incorrect regarding this L-DOPA? So, which of the following statement will be incorrect? Let me check the chat box. Again, I have, okay, I think I have two answers. Both are saying it is the number D. Let me check if number D is the correct answer or not. So, the correct answer will be Yes. Number D, the statement which says L-DOPA first decarboxylated by dopamine decarboxylase before entering the brain, it is false. So, levodopa, the chemistry of the amino acid levodopa is the biosynthetic precursor of the dopamine mechanism of action. In brain, levodopa is taken up by the dopaminergic terminals of the spectrum and converted to dopamine by the levo aromatic amino acid decarboxylase or dopa decarboxylase. Dopamine cannot be used since it does not cross the brain, enter the brain, crossing the blood-brain barrier. Levodopa itself is largely inert. Its effects depend on the increased synthesis of dopamine in the brain and are mostly related to the activation of D2 receptors. So basically what happens, so we have this tyrosine from levodopa is synthesized and levodopa can be converted to the dopamine. So when levodopa is it crosses the blood-brain barrier and then what happens? It is after decarboxylation it converts it into the dopamine and then this dopamine gets activates the dopaminergic transporters and which is and again related to the activation of the D2 receptors in the postsynaptic terminal of the striatum. Okay. So the statement which is number D, which is incorrect. Other statements, first, dopamine cannot cross the blood-brain barrier is correct. L-DOPA acts as a prodrug. It is also correct. L-DOPA can cross blood-brain barrier is also correct, and this statement is incorrect. Okay. Levodopa, it is not decarboxylated first. First, it crosses the blood-brain barrier. After that, it gets decarboxylated and converted to dopamine. Okay. Now, last question of today's session. It says that which of the following is not a primary pharmacokinetic parameter? Your options are number A, volume of distribution, number B, half-life, number C, clearance, and number D, all of the above. Which of the following is not a primary pharmacokinetic parameter? Volume of distribution, half-life, clearance, or all of the above? Which of the following is not a primary pharmacokinetic parameter? Let me know in the chat box A, B, C or D which of the following will be the correct answer. Which of the following is not a primary pharmacokinetic parameter? Which of the following is not a primary pharmacokinetic parameter? Volume of distribution, half-life, clearance, or all of the above? Let me check the chat box. Okay, only one answer I have received, which is saying it number D. Okay, let me check if number D is the correct answer or not. So okay, 1 second. Okay, number D is the only given answer. Let me check if number D is the correct answer or not. So the parameter which is not a primary pharmacokinetic parameter will be, it is number B, half-life. So basically, whenever a drug is given in the and the drug goes to the systematic circulation, after the administration, it goes to the distribution, and this distribution, volume of distribution is the first parameter here. And after the distribution, this drug goes to the metabolism and the excretion. Metabolism gives us the metabolized, excretion gives us the excreted drug. Elimination is basically metabolism plus excretion, and disposition is the distribution plus elimination. Okay, so primary pharmacokinetic parameters are basically the clearance and volume of distribution. Secondary pharmacokinetic parameters are half-life and bioavailability. So this is the pharmacokinetics. This is the amount of drug in the blood. His first is the absorption when the drug is taken. and with the time. Okay, so the absorption is the amount of drug in the blood is increased after the absorption, then elimination, and then after that the drug concentration gradually decreases. We have the Cmax, which is the highest concentration of drug in the blood. Then we have T half or half life, is the time when the drug has the lost half of its maximum concentration, and Cmin is the lowest concentration of drug in the blood. Okay, and this particular is known as the AUC or area under curve, which is the overall drug exposure. All these things we will discuss in the next class, okay, in the detail. Now when we will discuss about the drug administration, bioavailability and all, we will discuss in this our next class, because all these things are in detail in our week 7 course content. But I thought just to give it a little overview before before the next class. Okay, so this was the last question of today's session. So today what we have discussed, like we have discussed mainly the different metabolism. We have discussed the phase 1, phase 2 metabolism. What are the examples? We have seen a lot of examples of phase 1 and phase 2 metabolism, and I'm sure if you follow these things, it will be all the questions you can answer very correctly regarding this metabolisms. Okay. Then we discussed a little bit about the blood-brain barrier and all these things, the prodrugs, the various parameters. So today it was our week 6 live session, right? So now Cmax, let me just go to here. Yeah, Cmax will be the primary pharmacokinetic parameter. All these things, these pharmacokinetic parameters, I will discuss in the next class, okay, in detail, so don't worry. So I think the Cmax I have to check, because I think the Cmax it will for primary or secondary. I think I have to check it. Okay, the half life is the secondary, so I think Cmax, Cmin, all these things will be the secondary pharmacokinetic parameter, not the primary one. Clearance, volume of distributions, these are the primary pharmacokinetic parameters. This thing I will repeat in the next class also. Okay, I will check and then I will confirm you whether it will be a primary or secondary pharmacokinetic parameter. Okay, so I am sharing my email ID with you here. All these things, this pharmacokinetic parameters, the bioavailability and all these things we will discuss in the next class in detail. Okay. So this is my email ID. If you have any doubt, any query regarding today's class, you can drop an email to me. This class material, this presentation which I am presenting today, along with the video, this particular video, it will be uploaded in the NPTEL website. Where to find you? I have already mentioned you. You have to go to the course page. There you will find here the problem solving session. If you click to the problem solving session recording, you will get to redirected to a page from where you can download this PPT which I am presenting today along with this class recording. Okay. So today it was our week 6 live session. Now I will close the session for today. I hope if there is any problem, any doubt, please feel free to reach me through the email also. You can post your query in the discussion forum, and also you can let me know in the next class itself if you have any doubt or any query regarding today's class or any previous class. Okay. So I will close the session for today. Is there any issue or is there any problem you want to tell me? You can let me know now. Otherwise, I will close the session for today. So if there is no query, I will close the session for today. Thank you all for joining. We will meet on the next Friday from 4 p.m. for our next live session. Okay. I will close the session now. Thank you all for joining.